For this today's topic, we are going to deal with the basis of rDNA technology, that is enzymes in rDNA technology, otherwise called recombinant DNA technology. Boyer and Stanley Cohn proposed this technology. This technology mainly involves in the manipulation of genetic material in the organisms to obtain desired products or to develop a different characteristics in the living organisms. That is the recombinant DNA technology. Enzymes involved in rDNA technology, which is responsible for manipulation of the DNA molecule. The enzymes involved in rDNA technology are DNA ligase, reverse transcriptase, recession endonuclease, terminal transcriptase, nuclease, DNA polymerase, ribonuclease H, alkaline phosphatase, polynucleotide kinase. See one by one, what is the role, what is the function of each and every enzymes involved in rDNA technology. The first enzyme is DNA ligase. This ligase enzyme is isolated from E. coli and bacteriophage, and they are commercially used in rDNA technology. The main function of DNA ligase is to join the DNA fragments in the vector. It involves many cofactors like ATP or NAD+. Reverse transcriptase. It is used to synthesize complementary DNA molecule from mRNA template. It is also known as RNA-dependent DNA polymerase. It is isolated from retrovirus. This enzymes was isolated from retrovirus. Example for retrovirus is HIV. So they can make DNA from RNA molecule. Nuclease enzyme is one more important enzymes in rDNA technology, which is responsible for cutting the DNA at the sites which we want to know, which we want to clearly insert the DNA of any foreign origin. The restriction endonuclease enzyme mainly responsible for recognizing and cutting the DNA strands at specific sites. These site where the enzyme cuts is called restriction site, and they are isolated from many organisms, wide variety of organisms starting from bacteria to fungus. We have isolated more than 300 enzymes. The endonuclease enzyme mainly degrade foreign genome when enter inside microbial cell. But the host cell own DNA is protected from its endonuclease activity by methylation of the bases at restriction sites. Thereby, this enzyme, even though it is present in that microorganisms, but it cannot damage its host DNA because of the modification in the host DNA, which prevents the attack of this enzyme on its DNA. So this enzyme is mainly responsible for cutting any foreign DNA. Phrase. This is the enzyme which converts the blunt end of the DNA fragments into sticky ends. If the restriction enzyme cuts DNA, they will form blunt ends. The efficiency of the ligation is very low because the restriction endonuclease enzyme can cut the DNA by two ways. Either it can produce blunt ends or sticky ends. If there is a sticky ends means they can join together because of its complementary nature, whereas the blunt ends cannot join. So those enzyme which cuts the DNA and producing blunt ends, the ligation efficiency is very low. So the enzyme terminal transferase are responsible for converting this blunt end to the sticky ends. The terminal transferase enzyme, what they will do, they will synthesize a short sequence of complementary nucleotides at the blunt portion of the DNA so that the blunt end is converted into sticky ends. so that can be used for ligation very easily compared to the blunt ends. The next enzyme is nuclease. So this enzyme mainly hydrolyzes the phosphodiester bond on the DNA strand, which creates 3' OH group and 5' phosphate group. So we can say that the nuclease enzyme which breaks the phosphodiester bond in the DNA strands, thereby creating the 3' ends, free ends and 5' free ends. So it usually cuts DNA on either side of the distortion caused by thymine dimers or intercalating agents. The gap is filled by DNA polymerases and strand is joined by DNA ligase. So the nuclease, there are two types, endonuclease and exonuclease. Endonuclease means which can cut the DNA inside. Exonuclease means which can cut the DNA from the periphery ends to the inside one. Bases. The DNA polymerase is the main important enzyme which is responsible for forming DNA chain. So it is a very complex enzyme which synthesizes nucleotide complementary to the template strand. So they will add the nucleotides to the free end. It can be 3' OH end and help in the elongation process. It fills a gap when there is any repair mechanism goes on there. And also we have different types of DNA polymerases which is present in the prokaryotes and eukaryotes. In prokaryotes we have DNA polymerase 1, 2, 3 and eukaryotes we have alpha, epsilon, we have delta, we have gamma. So there are different enzymes for different functions which is mainly responsible in the replication process. Likewise, we have Taq polymerase. This is an enzyme which is obtained from Thermus aquaticus bacteria in the hot springs, and they are heat labile or heat stable, we can say that. Because of that, they are used in PCR. H. They remove mRNA from the DNA RNA hybrid. So H stands for hybrid. RNA is the ribonuclease enzyme which removes RNA from this hybrid molecule, and it is isolated from retrovirus. Likewise, alkaline phosphatase. The enzyme alkaline phosphatase, they are used to remove the terminal phosphate group from 5' end. Thereby, it prevents self-annealing of vector DNA soon after it cuts open by resection endonuclease enzyme. So the main thing, alkaline phosphatase, so it prevents again the 5' end to join with the previous ends. It prevents that joining process by removing the terminal phosphate group from the 5' end of the DNA molecule which is cut by the resection endonuclease enzyme. Otherwise, the 5' end of the phosphate group molecule will surely anneal with the existing ends of the DNA. A. The endoribonuclease that specifically degrades single-strand RNA at the C and U residues. It is a ribonuclease enzyme which can degrade single-strand RNA molecule at the two bases, that is C and U residues. So it cleaves the phosphodiester bond between the 5' ribose of the nucleotide and the phosphate group attached to the 3' ribose of an adjacent pyrimidine nucleotide. So because of this we are getting a cyclic phosphate which is correspondingly it will be hydrolyzed to form 3' nucleoside phosphate. It is also a type of ribonuclease enzyme which degrade RNA molecule, but it is unique compared to other RNases that it is a ribozyme, that it is not a protein. It is an RNA molecule which acts as enzyme. So they are responsible, the main function of these RNAsP is to cleave off extra or precursor sequence of RNA on tRNA molecules. So if there is any extra molecule or any precursor molecule present in the tRNA molecule that is removed by RNase P. Nucleotide kinase enzyme. This is responsible for adding the phosphate group to the 5' end after dephosphorylation by alkaline phosphatase. So it is a reverse of alkaline phosphatase enzyme. So alkaline phosphatase enzyme removes the phosphate group at the 5' end, but this polynucleotide kinase will add the phosphate group at the 5' end, thereby it promotes the annealing process. So these are the enzymes which is mainly involved in rDNA technology. Hope so. This will help you in the further lessons to know about the techniques and the usage of these enzymes in this technology. Thank you very much.